Okay. Hello, everyone. Welcome to the Ancient Genomics HEAS Seminar. And we're very happy to have Jenny Tung here. So Jenny is the director of the Max Planck Institute in Leipzig, Department for Primate Behavior and Evolution, since 2022. So that's a relatively new department there. And what she has been working in the field of primate behavior and genetics throughout her career with a PhD at Duke University in North Carolina in the USA, where she studied baboons' behavior and genetics, followed by a postdoc in Chicago, where she was also working on functional genomics and building up the portfolio basically to then start as a faculty at Duke University, where she's still co-affiliated, working on primate behavior and genetics in multiple different species. And she's also the co-director of the Amboseli field site in Kenya, which is one of the long-running primate study sites. And yeah, so we're very much looking forward to hear from you about what primates can tell us about ourselves as well.
Thanks, Martin. Okay. All right. Thank you very much for having me here in Vienna. Thank you, Martin, for the invitation. Before I start, I want to clarify that I think... There we go. I just want to clarify that I don't want to give you any false impressions here. I know this is the Ancient Genomics Seminar Series, and I'll talk a little bit about genomics. I will not talk about anything ancient. And hopefully that's clear from the title of my talk. I study living non-human primates, and I hope that we can find a point of intersection here in the sense that I think both ancient genomics and many non-human primate studies are motivated by the fact that within an anthropological context, it's actually impossible for us to understand everything we might like to know about our species by studying living humans alone. Okay. So what are studies of non-human primates good for? Here's a recently published phylogeny of about half of the species, half the extant species in the primate order. And I think it illustrates a couple of things. One, non-human primates are useful for comparative studies. They help establish the rules and patterns that govern evolution in our order. Do species that feed a lot on fruit tend to develop bigger brains? Is the evolution of the primate lineage heavily driven by our need to recognize and establish long-term relationships with other conspecifics and so on? A great deal of work, especially work that's been done at my institute in Leipzig, focuses on a very narrow comparison within primates. And that's this very, very specific one here between our little branch and our closest living relatives, the members of the genus Pan. So here we're looking at just the small distinctions that separate that one little branch from those around it. And third, and this is the area that I've probably spent most of my work on, is that other non-human primates allow us to gather information that's often difficult to gather when we're focused on our own species. They allow us to watch animals from the cradle to the grave at a sort of density of data collection that would be impossible to do in living humans. In other words, we can watch them day to day, day in and day out, and take very, very granular information on exactly what they're doing from moment to moment, both within and across generations. And that gives us information that is about the human phenotype that's impossible to gather from studying humans alone. All three of those perspectives are ones that we take in my department. Here's a collage of some of the species that we are currently studying. You'll notice there's even a few social mammals that are not primates that have snuck their way in there, mostly because of an interest in the evolution of strong degree of cooperativity outside of the primate order. Today, I'll focus my entire talk on one of them, and this is the species or set of species that I've spent the majority of my career studying. Those are the ones on the top left, and that is a baboon, members of the genus Papio. Specifically, I'll focus on baboons living in the wild in this single population. So this is the Amboseli ecosystem of Kenya. It's on the southern border of Kenya, right close to the northern border of Tanzania. I actually just came from there yesterday. And this is a set of our animals. Each one of them is individually recognized. And like I mentioned earlier, they're followed from conception, if we catch that, all the way till death or disappearance. These animals are particularly interesting as models for humans and for hominins more generally. They are large-bodied, mostly terrestrial, ecological generalists. They radiated in eastern and southern Africa and have spread throughout Africa and parts of Arabia to inhabit pretty much every ecosystem that you can find within the continent, with the exception of deep desert and some very, very deep rainforest. Okay, so actually, unlike a lot of the other great apes, these are generalists like us that manage to do very well in a wide variety of ecological circumstances. They're also highly social. They live their lives in obligate social groups, and they form individually differentiated relationships in those groups that can include both competitive interactions and affiliative interactions, what some people have called friendships that can last many, many years. I work on this population in particular because it has been the study, focus of long-term study for now 53 years, going up to about 10. We might be on our 11th generation of baboons. Since this study started, the population now includes about 2,000 or so individually recognized animals. So it's one of the largest long-term studies of wild primates in the world, and one of the longest running. I'm fortunate enough to do this through the foresight of Jean and Stuart Altman. Jean Altman is pictured here, who founded the site in 1971. They basically drove around Kenya in the 1960s looking for the best possible place to study wild baboons, and settled on Amboseli in part because of its abundant population of animals and because of the excellent visibility conditions. So you can see here it's actually very easy to follow individuals and keep close track of what they're doing. Today, the project is co-directed by Beth Archie at the University of Notre Dame in the United States, Susan Alberts at Duke University, and myself. So everything I'll be telling you about today is a product of that close collaboration. Oh, I wonder if I can, yeah, there we go, let's get rid of that. Okay. And of course, all the long-term data is collected through a much larger collaboration, especially with our long-term field team in Kenya. A picture of them from a few years ago is shown here. I want to particularly point out Kinyua Waratere, Raphael Mutatua, Langita Sayodi, and Sarah Sayalel, who are senior field observers and have been responsible for those day-to-day individually focused follows for the last 40 plus years. And in fact, one of the reasons I just came from Amboseli is because we were celebrating the retirement of Raf right here, this guy here, who has been working with the project since 1981. And so is probably one of the people in the world who knows the most about baboons that you could possibly find. So we'll miss him, but the project keeps on going. Okay. So I've split my talk into two pieces, so I can tell you two types of things that we learn about primate evolution with ramifications for our understanding of humans from both a sort of short-term perspective, the predictors of fitness within individual lifetimes, sort of the evolutionary ecology, life history, behavioral perspective. And then I'll switch over to the evolutionary genetic perspective, what we can learn about deeper time evolution, and particularly in the context of hybridization and admixture through genetic analysis. So I'll start with the first one. And if you forget everything else, I'll try to focus on two principal takeaways from our work in this area. The first is that although when we think about the components of fitness, there are two main components, right? There's the mortality components of fitness and there's the fertility components of fitness. We now know that it is variation in lifespan that explains by far the most variation in lifetime reproductive success in this species and likely in many other long-lived primates. So what I'm showing you here is a plot where each line is a different female with a completed life history in our population, limiting it to females who had at least one surviving offspring. So these are their reproductive life histories, and that's why the lines start at about 5 years of age, which is about the age of first birth. Each dot within a line shows when that female had a kid, had a live birth, which either survived to the average age of weaning, about 70 weeks in this population, or if it's grayed out, died prior to weaning. Here, I hope you can see that long lives make a big difference to how many offspring that females leave behind. Contrary to what used to be sort of the wisdom about natural populations, there is aging in this population. We do see increases in instantaneous mortality rates. In fact, quite steep increases as females reach older age. You can see changes in condition very clearly. This is the same female. This is Dottie when she was a young adult, and this is Dottie right before she died about 20 years later. And I think it's probably visibly apparent that there's some changes in her phenotype in that time. Dottie remains our record holder in terms of the longest lived animal we've known in the population. So actually, this line is her. And hopefully you can appreciate that because she managed to live so long, the number of offspring she left behind is considerably larger than the number of offspring left behind by these females who made it to reproductive adulthood, but didn't have the chance to produce very many offspring. This is the basic intuition behind why the mortality components of fitness are so important in determining who does well in this population and who leaves surviving lineages, right? Compare that, for example, to the difference in the age at first reproduction here. So you can kind of see that by the difference in ages on the x-axis when these females first gave live birth. There is variation. This is important to determining variance in lifetime reproductive success, but there's simply much less variation in it, and so it explains much less. Another major factor here is one that probably won't surprise you. This plot again only shows females with completed life histories who made it to the age at first reproduction. There's also what Alan Graffin called the invisible fraction. 61% of overall variance in lifetime reproductive success is explained simply by early life experiences and early life mortality. Many, many females don't survive to reproduce even once. And the sort of mortality curve this generates is very similar to the mortality curves that we see, different in sort of absolute scale, but very similar to the mortality curves that are described for human populations. So this is another way of showing you what I just told you. This is, again, females with completed life histories based on age at death, starting from sort of the minimum age at reproductive maturity, about 4 years of age. And this is the number of surviving offspring. There's variance in this plot, but basically a female baboon in Ambacelli produces another surviving offspring every 2.1 years. And that line, that simple line, describes most of the variance. So the mortality components of fitness explain overall about 90% of the variation in LRS in this population. And there are non-zero contributions of fertility-related traits, but they're small. And this actually matches, although it's a nice empirical example, I think, with the expectations for a long-lived species like ours. I think that suggests, for example, that signatures of selection we might see in the human genome over time probably, if you had to place a bet on it, have more to do with mortality than with fertility. And it also raises some interesting questions about the focus on fertility in most long-term field studies. I actually think that generally is because fertility-related traits are easier and faster to collect than survival-related traits. So this takes many, many decades before you can produce plots like this, as simple as they are. The other thing I want to tell you is, of course, if we know that mortality is what's important, or what's perhaps most important, a natural question is, well, what explains mortality? What explains variance in how long individuals live? And one of the other patterns that has emerged quite clearly from our work over the last decade or so is that the social environment, and particularly affiliative social relationships, are among the most important predictors of survival in species like these. So social relationships in the baboons, in case you don't really watch or think about non-human primates, are mostly solidified by interactions like these. This is a grooming interaction. This is Rwanda is the female being groomed, a long-lived female in our population. population who just died in December of last year by her then adolescent daughter, Rodeo. Rodeo actually just had another baby last week. I got the chance to name it while I was there in Kenya. And they are engaged in the primary glue that establishes social bonds in this species. Our first attempt to describe what these social bonds might do, or at least might predict in this population in terms of survival, came in 2014, where we looked at basically the frequency with which females groomed other females and the frequency with which females were engaged in grooming interactions with adult males. We don't show male-male grooming relationships here simply because in the particular species that we study, males don't groom very much together. This is not true in all species of primates, but that's why they don't show up here. This is the result of that analysis, again, focusing on adult survival only, so conditioning on females who survived to maturity. That's why this line starts here. These graphs show the survival probability estimated from our models of those individuals who sort of engaged in these affiliative interactions at about the average rate in green versus those that were consistently, or those that would be predicted to be consistently most top quartile of social connectedness. They're doing the most grooming versus the lowest quartile of grooming relationships, or the most socially isolated animals. You can see here that there's quite a big difference actually in expected median survival for individuals who are strongly connected versus those who are weakly connected. Interestingly, you might think that these traits are correlated. They actually are not. There are two paths to social integration in this species. For females, you can form very strong or frequent affiliative interactions with other females, or you can do that with males. And those two traits are almost completely uncorrelated. If anything, they're slightly negatively correlated, suggesting perhaps a time or resource trade-off between same-sex and opposite-sex relationships. What that means is that if we put those together and we estimate what median lifespans might look like for the females who are most socially connected to both sexes versus least socially connected to both sexes, we end up with a difference that is quite large. This is these lines are approximately median survival, and you see a difference of about 4 or 5 years in life expectancy. And remember, we can translate that back into surviving offspring at a rate about 1 surviving offspring every 2.1 years. So this is a significant difference in terms of thinking about fitness outcomes for these animals. More recently, we've been interested in what about social relationships leads to these kinds of outcomes. There are a variety of ways that you can measure how socially integrated or how socially isolated an animal is. You can do the same thing for human populations. You can think about the frequency of affiliative interaction. So that's the result I basically showed you earlier. In this plot, this sort of yellowish dot in the center is the focal, and you can imagine that individual grooming with a different number of grooming interactions with a number of different social partners, leading to a particular value for just how much you engage in these kinds of behaviors. You can also think about frequency and that's weird, quantity in a different kind of way by just sort of counting up the number of social partners a female might have. And then you get a different number with a different sort of amount of variance in the sample. So same social network, but summarized in a different way. Joan Silk and others have emphasized that perhaps frequency doesn't matter so much, but what matters are your closest friends. That if you think about things like coalitionary support, where your social partners might be, putting something on the line to provide support for a particular focal, that it is the strongest, most consistent, and most reciprocal relationships that matter. In other words, measures of relationship quality instead of quantity. So here, the pictures focus on the 3 strongest social partners for this focal individual instead. And you can see again here, this changes the numbers and it changes the relative ranking of the individuals in the network. And finally, others have argued for sort of social network-based metrics, including centrality in the network. So maybe what matters is not only the individuals that you interact directly, but also how they interact with the network. If you removed a particular individual from the network, how much does that perturb the structure of the social network overall? Interestingly, this has been described in human populations as well, actually long before this became a major topic of study in non-human primates. These quantities are somewhat correlated, but actually not very strongly correlated, especially quality and quantity measures. And some folks have actually proposed that there may be direct trade-offs between investing in many, many social partners versus investing in strong relationships with a few. Fernando Campos, who's a former postdoc with Susan Alberts, has been taking a look at this in some work that we haven't yet published yet to ask, if we look over an entire data set for everybody that we're able to include, which of these things actually seems to matter most? And here on the y-axis, this is a measure of mortality risk. Basically, the further down it is, the lower, the bigger of an effect size the predictor has on the top. And what you can see here perhaps already is that what seems to matter most is actually these measures of quality, at least the baboons, at least in this particular population. And in fact, you can see that through model comparison approaches as well. It's not that other measures don't pick up this sort of survival signal. It's that those strong bonds seem to be the most predictive of mortality in our population. How many bonds do you need if you're thinking about your own life and how you want to spend the rest of your day? What it looks like to us is that it's a relatively few strong social bonds that matter. Somewhere between 2 and 4 of the strength of your bonds to your strongest social partners, if you are a female baboon and Amboseli, have the largest predictive power on survival in any given year of life. I told you that early life also matters quite a bit. Many individuals don't survive past early life. There's high mortality in the first year of life. We also have come to believe that experiences during early life, and especially relationships, another form of social relationship between kids and their mothers, matters quite a bit to the long-term outcomes, the sort of life course outcomes for these animals as well. And this is the sort of thing that is detectable not only in instantaneous mortality risk early in life, but also in the long-term consequences of the experiences that infant baboons and juvenile baboons face before they become reproductively mature themselves. A number of years ago, inspired by some work on what's called the adverse childhood experiences model in humans, we decided to look at this through simply summing up the number of serious adverse experiences that baboons might face early in life. These are the 6 that we consider, and most of them are mediated by a young baboon's mother. So the social status of an animal when it's born, mediated by its mother, whether or not that mother was present throughout the infant and juvenile period, that is, whether mom survived until an individual turned 4, sort of again the youngest age at their own reproductive maturation, the social integration of that mother, whether she turned around and had another offspring soon after the focal individual was born. So this is based on an interbirth interval of 1.5 years. That's approximately the lowest quartile in our population. And also 2 indices of sort of ecological resource availability and competition. So the density of the group upon the birth of the offspring, and also the amount of rainfall in the first year of life. This is a highly variable environment, and sometimes can go into droughts that approximate the sort of desert southwest of the United States. So we did something that is fairly abiological, and we followed the example set by human social science researchers, and we sort of just binarized all these, and we counted them up, and we asked whether that predicted adult survival. So not the immediate death of an infant or a juvenile, but its long-term outcomes later on. I probably will never get as clear of a result again in my career. This is something we actually had not noticed before we added up our data in this particular way. Let me walk you through it. This pie chart basically gives you the distribution of adverse early experiences faced by female baboons in our population. About 20% of them are animals we think about as our silver spoon babies. Nothing particularly bad happened to them early in life. They had a pretty advantaged first 4 years. Another third of them experienced one of those sources of adversity. They lost their mother. They were born to a low-ranking family. They were born to socially isolated family, or so on. Another quarter experienced 2, and then there are these few animals who just got, sort of battered by repeated sources of adversity when they were young. We only considered females who survived to adulthood. So this is adult survival stratified by whether they experienced 0, 1, 2, or 3 or more sources of early adversity. And you can see this actually just enormous difference in how long they live. Those individuals who experience 0 or 1 source of early adversity, they live conditioned on reaching adulthood, a median of about 19 years. Those who experience 3 or more have lifespans that are truncated by about a decade. Okay, and if you're trying to put this in human years, you can kind of think, is this, I don't know if this is a thing in an Austrian context, like a dog year is 7 human years? Yes, okay. So a baboon year is about 2 to 3 human years. So if we translated this into sort of expected lifespan terms in humans, we're talking about an effect size of about 20 to 30 years of shortened adult life. And again, if you think about this in the currency of fitness, in terms of surviving offspring, that's quite a large effect on average in terms of the likelihood that a female will leave, copies of her genome behind in her descendants. There's actually an even bigger effect than we originally realized. Matthew Zippel, who's a former graduate student with the project, advised by Susan, was interested in whether those females who lost their moms grew up and experienced lower offspring survivorship as well, controlling for those offspring's own environment. Okay, does that make sense? So before we were looking at what happens to this individual, now we're asking what happens to the offspring of that individual, controlling for whether it had, a mother around and was high ranking and then lived in a drought and so on. And what we find is that these females grow up and experience reduced offspring survivorship, right? So females who experience maternal loss when they themselves were kids have a harder time raising their own offspring to reproductive maturation. So the sort of evolutionary consequences of that sort of severe form of adversity early in life ramify forward in time to intergenerational consequences. I won't talk too much here about the mechanisms through which this appears. We don't think there's anything magical here. We think it's probably just a condition effect on this F1 individual. But regardless, it suggests that something that might have happened 2 decades ago or 2.5 decades ago continues to be observed through the life history of the animals that remain in our population subsequently. Okay, so 2 overall patterns that I think are takeaways from the first part of my talk. What predicts fitness within individual lifetimes? We need to pay a lot of attention to things that explain variance in survival and in mortality. And of those things, social relationships and the social environment are one of the most important, especially conditioned on reaching adulthood. And in addition, early life experience, especially those relationships between mothers and infants, are particularly important in whether individuals make it to adulthood and then even subsequently across time. And those are things that actually fit very well with data from the social sciences in humans, where in modern populations, social integration has long been described as one of the primary predictors of mortality risk. This is actually old data from a study in Alameda County, California, published in the 70s. Where there's follow-up of adults stratified by whether they were more or less socially integrated in their network. Individuals who are more socially integrated survive longer, both women and men. And also the effects of early life adversity. So this is one of the adverse childhood experiences studies that I mentioned earlier. This shows mean age at death. Again, for American population followed in Southern California, and you see these very, very large effect sizes as well. In the social sciences literature, this has long been interpreted as partially an outgrowth of the sort of modern human environment, which changes the way that we socially interact with each other and changes the type of environments that children grew up in. I think our work and others is suggesting that these are actually predictors that have long been important in the primate lineage, far predating the evolution of humans specifically. And in fact, if we look at the data so far, it's a limited set of things because these have to be quite long-term studies in order to get the mortality data. But if we look at the data so far on social relationships in natural mortality populations of social mammals, in fact, we do see this repeated pattern where one of the major predictors of survival has to do with measures of affiliative social relationships. And so this is from a review, so it's actually a little bit out of date now, that we put together about 4 years ago. And you can see it's a very common pattern so far in the primate studies that have been done. So this is from social epidemiology. This is the fundamental cause theory that argues that social factors, socioeconomic status, competitive social relationships, and social support should be treated as what the authors called fundamental causes of disease, things that explain all kinds of different disease outcomes through many different mechanisms. And so perhaps one of the most important things to measure when you're interested in the health of human populations. We might perhaps ask whether this is also true in social mammals, including many social primates, when we want to think about the determinants of fitness over time. Okay. So now I'm going to switch gears entirely. So I can wait till the end for questions, or I can take questions on this part now as you want. As long as Maeve says it's okay.
Okay. So, what is you have a real bulk of data available? And, what is the median interbirth interval in that group?
Median interbirth intervals for individuals who have live infants is 2.1 years.
2.1.
Yeah.
It's interesting. And about the grooming collections, which individuals are preferred to be groomed? Related individuals or individuals? Because as you talked about friendship, and Mats had written about it in detail stuff. But anyway, do they change with age their relationship to group members? The frequency of grooming, for example.
Yeah, so they do. So I'll answer the first question first, because which individuals groom more or what type of dyadic relationships form depends on whether you're talking about same-sex relationships or opposite-sex relationships. So for same-sex relationships between females, grooming partners, preferential grooming partners are close kin. Your mother, your kids, your sisters. In this particular species, females are philopatric and males obligately disperse. So females tend to form quite long-lasting and close bonds with their female relatives and less so with their male relatives. So kinship is the most important. And so what you do get is change over the life course, where at the very oldest ages, the rates of grooming decrease a bit. But in addition, the pattern of kin that you have in your group changes as you age, right? So when you're young, mom is most important. Mom is almost always the first, well, not almost always, but most frequently, by far, the first grooming partners for animals, and remains the major grooming partner for some time as the animals grow up. Eventually, if you're a successful female, you're going to replace that with your own offspring.
Successful female means, for example, high-ranking lineage.
Oh, successful female, and I just meant if you survived. If you're still around, and then if you had kids. Okay. For opposite sex relationships, interestingly, the clearest determinant we can see is actually rank. So high-ranking females are making friends with males, and they tend, on average, to be high-ranking males. And it's one of the reasons that we increasingly think that those males are a resource that females basically compete over. Yeah.
Hi, just I want to ask a question. I don't know because I don't think it will be covered in the next part, but anyhow, it's very interesting. But I'm just keep on thinking about epigenetics because I was thinking about longevity, epigenetic methylation clock.
Yeah.
Females, especially up to 3 generation inheritance, so to speak, and especially as we know from humans. I don't know if you've done also with primates that like emotional or could be physiological stress or emotional stress certainly has an impact on the methylation clock, and therefore potentially I would imagine could also be a co-founder, for example, perhaps for longevity. If you can say.
So we've done quite a bit of DNA methylation work on the population. We published a paper on a baboon DNA methylation clock a few years ago and looked at predictors of the clock based on the sort of socio-ecological variables that we think are important for life history in the population. The only thing that was correlated with sort of older than expected for age or younger than expected for age in our clock, which predicts actual chronological age quite well, was actually male dominance rank. And I'd be happy to talk about that in more detail because that actually corroborates other evidence that we have that individuals who compete successfully, males who compete successfully for high rank are experiencing some physiological wear and tear. In general, I would say that we also actually published a paper last week, so thank you for letting me advertise it, on those different sources of early adversity and their signature in the DNA methylation of individuals that we followed into adulthood. And I guess what I can tell you very quickly about that is that the major results are that it is the sources of adversity that we can most clearly link to resource deprivation, like being born in a drought or being born in a very bad habitat where food is likely much more limited, that correlate much more strongly with methylation signatures that appear in adulthood, and actually less so the sort of social and maternal offspring dynamics that I focused on today. And that typically that signature is it's actually very weak, other than when you get multiple sources of sort of resource deprivation together. So that's out in PNAS last week, and I'm happy to talk about it more if you'd like to. Yeah.
Thank you very much. Thanks.
And you're right, I'm not going to talk about it all next. So yeah, good question.
Okay. Yeah, I mean, I guess we can continue with the second part.
Sure. Okay. So the other thing we can do with living primates is think about how to reconcile the signatures that we see in genetic or genomic data with the observational data that we have on their behavior and life history. And that's something that is obviously of great interest to anthropologists. Here's a fictionalized artist rendition of what maybe it looked like when the ancestors of modern humans met other archaic hominin groups. You can see that guy in the background saying, stay back, stay back. There's these other people over there. Who knows what they're like? You can imagine that was a part of it, sort of defensive in-group, out-group relationships. You can imagine that in some ways they might have gotten over that, since we now have ideas about how gene flow happened multiple times, or maybe that was an aggressive encounter in and of itself. The fact is we don't really know, right? This is a highly, highly behavior-focused rendition of those encounters. And as one of my former graduate students always like to say, well, behavior is one of those things that doesn't fossilize, right? Echoes of behavior can fossilize, fair enough. But the level of detail that we might want to know about what happens when two diverged taxa interact is one that's actually very, very hard to recover in the history of our own species. Okay, because we just don't have any other representatives of our genus left alive. It turns out, though, that admixture and hybridization, including in natural environments, is extremely common in the extant primates. Take these estimates with a grain of salt. It was my attempt a few years ago to try and provide a general estimate for the proportion of recognized extant species that are known to hybridize in nature through field studies or other kinds of reports based on the sort of larger taxonomic lineage lineage in which they are members. For groups like baboons, the numbers are really quite high. 30 to 40% of species are known to actually hybridize with other members of the same or sometimes even different genera. This is, in fact, particularly well studied in the species complex that I work on. So these are the 5 of the 6 currently recognized species of the genus Papio, which is divided up into this sort of northern clade and this southern clade, including its outgroup. This is the gelada monkey, Therapithecus, approximately 3 or 4 million years diverged from the other baboons. Those blue lines represent cases of inferred ancient admixture, and those red lines all represent cases where there is an active hybrid zone that has been studied in living populations, or in the case of these hybridization events with gelada, a witnessed case of hybridization reported in the literature from the wild. This is true despite the fact that these live in actually fairly clearly geographically separate ranges within Africa, and the areas where you can find hybrids are quite restricted to the sort of the meeting points between those ranges. And so when we think about our population, it's interesting to ask whether hybridization and admixture has had an impact on the evolutionary dynamics of these animals. So this is sort of the timeline of our field study. This is a picture of Jean and Stuart Altman in 1963 when they were out scouting in Kenya. And they wrote a book about that initial reconnaissance throughout parts of Kenya, and described what they found when they got to the Amboseli area. They said there's no disagreement that the baboons that we studied in Amboseli should be classified as yellow baboons or cynocephalus baboons, Papiocynocephalus, the scientific name. A number of years later, about 15 years into the long-term field study, actually, I guess the first immigration event was observed in 1982, so about a dozen years, Jean and her student Amy Samuels reported the first observed immigration of a Papio anubis, an anubis or olive baboon male, into this population. That male integrated into the existing social groups and was successful in having offspring, establishing what they described as an anubis cynocephalus hybrid zone in this region of Kenya. That produced individuals who look a little bit like this. This is a male who lived in our population named Dubu. It's based on his pedigree. He is an F2 backcross to Anubis, so we would have estimated his genome composition to be 75% Anubis, 25% yellow. Fast forward to a few years ago, and we can resolve what's going on in the population much better now because we're able to generate genome-wide resequencing data. And so this is the result of our attempt to resequence basically every individual in the population who we knew and who we had high-quality samples for, so blood or tissue-based samples collected through periodic dartings or from corpses. So each of these plots is made up of a series of lines where each line represents a single individual sampled in Amboseli here or in neighboring populations, including from captivity, where we think we know where the original founders of that population were sampled from. Each line is about 100 megabases, I think, of one chromosome, and they're colored based on our estimates of the local ancestry of each of those individuals in each of the populations. So you don't have to go very far to the west. There is the Great Rift Valley here before you get to populations that look to be pretty clearly unadmixed or close to unadmixed Anubis. And you don't have to go very far south. There is no clear geographic barrier here to just so you're aware, before you get to populations that look both phenotypically subtly different and genetically like they're relatively unadmixed. And then you have Amboseli, which looks like this. It's full of individuals sampled from birth dates that range from the 60s up until the early 2000s, maybe mid 2000 teens here. when you see a real mosaic of ancestry, including cases where you see homozygous local ancestry across large tracts of the genome, and cases where you see primarily yellow ancestry, but with periodic introgressed segments from Anubis individuals. So in all of these cases, green is homozygous Anubis, orange is heterozygous, one copy from Anubis, one copy from yellow, and yellow is homozygous yellow. Okay. We can actually look at the temporal change in the composition of the population going back into the 1970s. On the y-axis, you can see mean genome-wide Anubis ancestry, the minor parent ancestry in this population. And this is plotted based on who was living in the population in every year of the study going forward to, I think, about 2019 or 2020. The Anubis genetic composition of the population is clearly being brought in by male immigrants. You can see that immigrants are on average more Anubis-like, but also hybrid than individuals who are born in the population. And consistent with what Jean described in the 1980s and then some updated results from the early 2000s, that proportion has been increasing over time. Okay, you can see this upward trend. That increase is probably driven by a few of the ancestry-related phenotypic differences that we're aware of in this population. So males who have more Anubis-like ancestry tend to start their independent reproductive careers earlier than males who have more yellow-like ancestry. So they leave their group of birth to seek out reproductive opportunities in other groups at a somewhat earlier age. We know that both males and females who have larger proportions of their genome from Anubis ancestry tend to groom with each other more often. They tend to form stronger opposite sex social relationships, and they tend to do so somewhat assortatively as well. And we also know that males who have more Anubis ancestry tend to have a subtle but detectable advantage in mate competition. So most conceptive events in this population happen during these sort of 5-day windows when females are cycling, and males tend to try to monopolize those females' time. At that time, what is called mate guarding in most species and in baboons for historical reasons is often called consortships. Males who have the opportunity to consort a cycling female, including a conceptive female, tend to have a slightly elevated Anubis ancestry, controlling for a lot of the other things that really matter for mating this population, like dominance, rank, and so on. Yet one of the things you might have noticed here is although this plot goes back until the 70s and early 80s, that first Anubis immigrant, which I told you was observed in 1982, entered a population where, if you look on the y-axis, actually was not so much of an unadmixed yellow baboon population. With our genome-wide data, we can now estimate that even in the 70s and early 80s, there are significant amounts of Anubis introgression already in this population. That probably accounts for the reason that if you look historically, you probably never had reason to do this, into the taxonomy of baboons described over time, the animals in Ambicelli were described as a slightly different morphotype of yellow baboon than the animals that you would find further south or further towards the coast. Okay. So we actually have in our population a mix of individuals. We have those that, based on pedigree records and various genomic criteria, we think have had no very recent Anubis baboon ancestors. Those are animals like Rongi on the left, and you can see her local ancestry painted across her genome, where she definitely has introgressed segments of Anubis ancestry, but they're not so long, and they rarely occur in a homozygous state. Versus animals like Eclipse, again, we know both of their pedigrees, where we know she had an Anubis or Anubis-like ancestor fairly recently in her lineage. And you can see the composition of her genome also looks quite different accordingly. So what we have is a situation where I've described a variety of phenotypic correlates of ancestry in this population that suggest an Anubis ancestry advantage. Right? They're maturing a little bit earlier. They have some advantages in terms of mate competition. They form closer social relationships with each other, or at least they tend to groom each other more. And I've already told you that's a predictor of mortality in this population. And yet, the range where you can find hybrids is really small. You can go 30 to 50 kilometers in either direction outside this hybrid zone. They look phenotypically like pretty good Anubis or phenotypically like pretty good yellows. And some of our preliminary genetic analysis from microsatellite loci supports that as well. And we seem to be in a situation where there have been at least 2 and possibly multiple waves of admixture coming into this population or to animals who have moved into this population subsequently, suggesting that despite the fact that hybrids are clearly fertile and viable in our population, there's something that's maintaining taxonomic integrity. There's something that's keeping them apart. To ask about whether we can see echoes of that in the genome, we were very inspired by the work that had previously been done to look at isolation between the ancestors of modern humans and other archaic hominins. So I'll just describe 3 patterns that suggest to us that indeed there is selection against introgression in this population, contrasting in each case what we find in baboons with what's been described in the case for humans. So one is just to look in segments across the genome for regions of the genome that are more diverged versus less diverged, right? So these are, I think, 250 kb chunks in this particular plot, and you can count the number of fixed differences between, say, humans and Neanderthals in one case, or Anubis baboons and yellow baboons in another case, okay? So there's the human Neanderthal case, and here's the baboon case. And in both cases, you see a reduced level of introgressed ancestry for regions of the genome that are more diverged at a sequence level, suggesting likely the presence of more incompatibilities. You also see very similar patterns between the human archaic hominin case and the baboon case. If you look at patterns of introgression with regard to region-specific estimates of linked selection or background selection. This is using the B statistic developed by Graham McVicker and colleagues, which basically translates to linkage to regions of the genome we think are probably constrained, like exons and promoter regions. We also see reduced levels of introgression in putative enhancers in the baboons as well, again, echoing a pattern that's been described in humans. And finally, Molly Schumer and colleagues described a pattern where introgressed ancestry, in this case, both in the Neanderthal case and the Denisovan case, is reduced in regions that have higher recombination rates. The intuition here is for elevated in regions that have higher recombination rates. Sorry, I have to get the direction correct, and it's easy to switch those in the language. So the intuition here is that for regions where recombination rates are very, very low, an introgressed variant that is perhaps mildly deleterious will, through selection against it, remove a bunch of the chunks that surround it as well, whereas regions that have high recombination rates will tend to allow some of those sort of more neutral regions to stay, even if there are variants that are selected out. And again, we see very similar patterns in the baboons, as has been described in the case of humans and archaic hominins, suggesting in some that there is, in fact, something going on that selects against free gene flow between those baboons. I will say again, we know in our population that we have a combination of what we call sort of historic hybrids, individuals in this distribution on the left-hand side who have Anubis ancestry, but are the products of hybridization events that are probably quite far in the past, hundreds to thousands of years ago, perhaps. And individuals who we know have very recent Anubis ancestors, many of whom we actually knew as part of our study population. And these patterns are very, very much driven by the historic hybrids. They attenuate quite a bit when you look at the patterns of ancestry from those recent hybrids in the last few generations. However, even so, the number of fixed differences in chunks of the genome and the local recombination rate does predict weakly, but it does significantly predict a low frequency change in our population if we look at the temporal sample, like what the frequency of Anubis versus yellow fragments are in the genome over time from the 1980s to the present, suggesting that whatever it is, whatever the phenotypes are that are restricting free gene flow are still acting subtly in our population today. We reasoned that all of those patterns imply that there's something functionally important about the segments of the genome that are being selectively removed. One of the other things that we can do in living primates that's quite difficult, as I understand, to do with things that died a long time ago, is to look at phenotypic variation in the current population at a molecular scale. And we have done that for quite some time by measuring gene expression patterns in the animals in our population. So this is just sort of a picture of how we do it. This is Kino Wariteri, one of the people that I introduced to you earlier, very, very early in the morning, carrying a meter-long blowgun. So we dart our individuals through breath-propelled darts that contain veterinary anesthetic. When the air is very still and nobody is looking, then he'll simply raise that blowgun and dart our target, who we can then draw blood from. Okay, so using patterns of gene expression that we've measured for a number of animals in our population over the time, we identified regions in the genome where ancestry predicts the level of gene activity, the level of gene expression, so local ancestry expression quantitative trait loci. We reasoned that if we look at the distribution of ancestry EQTL effect sizes, that those that are larger might be most restricted from introgression compared to those that basically don't exist or are the smallest, right? So that there then basically selection against perturbation of gene expression that has diverged between these two populations or these two species. And in fact, that is what we see. So if we look at the top 10% versus the bottom 10% of effect sizes, or the top 15%, 20%, or so on, you can see that there's this difference in the amount of minor parent ancestry and the amount of Anubis ancestry in those particular regions or genes of the genome. You can also ask about this in correlation with local recombination rate, and we see a stronger relationship between recombination rate and minor parent ancestry for regions in and around genes with strong local ancestry EQTL versus genes that have little to no evidence for ancestry effects on gene expression. Okay. So this dovetails with hypotheses that have been put forward in the archaic hominin case that has said, well, you know, if you look at modern humans, archaic introgressed regions are no more likely to perturb gene expression than variants that probably arose in modern humans. Maybe that's because the ones that had bigger effect sizes after divergence have already been weeded out by selection. I think our data are consistent with that possibility. And finally, just briefly, the other thing we can try and do is try and understand what the phenotypic mechanisms of isolation are. And we don't have nearly as much traction on this as I would like to. But of course, one place where this might pop up is in the context of mating. So this is an adult male baboon consorting a cycling female. When I was a student, I did an analysis of consortship probabilities in this population that suggested a pattern of subtle assortative mating, where males of a given genetic ancestry, this is most Anubis-like here, this is most yellow-like, were more likely to successfully consort with females who had a similar ancestry pattern than them. So my former student Ariel Fogel took that suggestion and decided to run with it by asking about whether or not that might produce long-term patterns of linkage disequilibrium that could not be explained by sort of simple physical linkage in the genome of the animals in Ambicelli. So basically what we're talking about here are cases where, say, across two chromosomes, you see matched patterns of ancestry, Anubis ancestry at this locus, Anubis ancestry at this locus. That could be by chance. If you look at it across many individuals and you tend to see matched ancestry at these loci and rarely see mismatched ancestry, that may be a signature of reproductive isolation. This is logic that's been used actually in a number of different hybridizing species, swordtail fish, mice, Drosophila, and so on, mostly in the context of looking for Dubjansky-Muller incompatibilities, so post-zygotic incompatibilities. But this is a pattern in the genome that can also arise if individuals are assortatively mating, and so individuals, so variants in the genome that have something to do with the cues they're using for assortative mating sort of co-segregate like this. So she looked at partial correlations, pairwise partial correlations of ancestry genotypes, basically homozygous Anubis, heterozygous, homozygous yellow, for markers across the genome, and looked at the ones that were most extreme, the 99.99% most extreme patterns of positive ancestry correlation. And then she asked, are these things explained by assortative mating? And here again, we go back to the actual phenotypic data from the population, where we have a lot of information on who mated and who didn't, given the opportunity to do so, right? So the outcome here is whether an adult male and an adult female mated, when they were living together in the same social group, when that female was potentially conceptive. This is about 2,000 or so different estrus cycles recorded over time, controlling for, things that affect inbreeding avoidance, social hierarchy effects, age effects, and so on. So the basic question here is whether individuals with matched ancestry at these really extreme ancestry correlated pairs are more likely to have in fact mated than individuals who are mismatched for ancestry state. And this is what she finds. 92 locus pairs involving 131 different loci have a significant correlation with mating behavior to 10% FDR in our population. They're shown here in those red stripes. Interestingly, those particular mating-associated variants seem to have lower representation of Anubis ancestry in the population than you would expect by chance, and also lower than you expect from just the overall 99.99% extreme ancestry correlated variants that she tested in the first place. We don't have a very clear idea of what these things do, but interestingly, at the same time, in work that was completely independent from us, Graham Coop, Pavitra Maralhar, and Carl Veller did an analysis arguing that this type of assortative mating could actually speed the purging of introgressed variation from genomes, simply because assortative mating sort of has this bundling effect to increase variance across individuals in the ancestry that they carry. Based on the mating data from our population, they argue that if Anubis ancestry is in fact deleterious in some way, then the estimate of the observed correlation in ancestry between mating pairs would translate to an approximately 20 to 24% accelerated purging of Anubis ancestry, which is quite nice, but presumes that Anubis ancestry is in fact deleterious. This is the closest evidence, and it's unpublished because we still need to poke at it, but this is the closest evidence we have so far for those deleterious effects. And in fact, again, it's quite subtle relative to what we observe in the population in general. So what this is survivorship to age 4 of offspring from mothers of different genome-wide ancestry, where it looks like those who are in the most intermediate level tend to have slightly, subtly reduced offspring survivorship relative to background. Whether that's enough to drive those sorts of patterns that we see is still an open question, but provides at least the first glimpse of some of the things that might be guiding the genomic analyses that we observe. So I'll just wrap up and say that I hope I've convinced you that there's reason to study living primates, even though, of course, they don't replicate the exact patterns that we care about in humans, in order to understand the sort of phenotypic dynamics that might explain sort of signatures of selection against introgression. We also see signatures of selection against introgression, and interestingly, they are similar qualitatively in strength to what's been observed for archaic hominin introgression in humans, but are completely compatible with animals that look like they're doing just fine in our population, unless we get to very granular analyses of phenotype. So I think that provides a note of caution, perhaps, that the sort of data that we have from genomes alone will never be sufficient to tell us what phenotypically is happening when two groups meet. And so I suspect that humans and Neanderthals, I don't know this, and I hope that work by folks like people in this room will tell us over time, may not have actually been at the very, very brink of separation, at least when you put them in the comparative context that non-human primate studies can bring. It turns out that some of these animals are being selected against, even when to our eyes they look like they're doing very, very well in the population. And so with that, here's a picture from Ambaselli from Friday morning. And I just want to thank you for your attention to those two very different segments of my talk. And if there's time, I'm happy to take any questions.